Hi, my name is Duane Johnson. This is my lab assignment, Aseptic Technique in Culturing Microbes for week 2 at Los Angeles Pacific University for general microbiology. So first off, we're going to be starting with inoculating some E. coli. So let's begin. We're going to grab our nutrient broth with our sterilized pipette. I'm going to sterilize the tip of the nutrient broth over the flame. So I think that's sufficient. I think I melted my tip. So I'm gonna pipette out 0.25 of the nutrient broth. I'm gonna grab our E. coli tablet. I'm going to try not to melt this one. I'm gonna add the nutrient broth. Don't think I quite got enough, so I'm just gonna grab just a little bit more. Careful not to touch the tip with the pipette, as well as the tip of that to keep it sterile. So careful not to touch the tip of this as well. So right in here, we're gonna add just a little bit more, just so it dissolves really well. Now, we'll go ahead and place this back in here. We'll draw it back up, sterilize this. Give this a good shake. And at the same time, I'm gonna empty out the pipette after it was sterilized and sterilize it once more. So I'll do this simultaneously. Then afterwards, after this is dissolved, we'll add this culture into our nutrient broth. Not dissolving very well. So here we are finishing up the E. coli culture with the nutrient broth. Still shaking. It's about almost all dissolved. But so far my mistakes were I placed the nutrient broth too long under the flame, so I kind of melted the cap. so I might run into some problems there. Hopefully it still caps nicely so it doesn't have any air going into it to possibly contaminate and ruin the culture. And then also I re-sterilized the pipette. I guess that's not too bad. I made sure it was dry before I recultured, or before I'm going to reculture, but it's sterile. So far those are my mistakes. Now we are going to transfer our E. coli culture into the broth. So it's pretty well dissolved. Now, I'll just go ahead, place the cap. We got a bit of a microbes right there at the tip. So I shouldn't have done that. I should have squeezed prior to putting that in there. So we have a little bit right here. Go ahead and place that in. So I'll just leave that squeezed and get the rest of this as much as I can. So hopefully that will be enough. I'll just reflame this to sterilize it. That'll do. Place the cap back on. It's a bit tricky with these small caps. Set that in the workspace. Now, I'm going to take my pipette, squeeze it a little, back into the alcohol to sterilize it, fill it up. Now we'll have to reflame the E. coli nutrient broth with the culture in it. Just quickly, this time I'm just going to quickly pass it over a few times. Then wait for it to possibly cool a little bit. And then see if it caps well. Well, got a good seal, so I hope this will be fine. We'll place it right back into the working space. Now we're going to do the same with the Staphylococcus E, or Staphylococcus epidermidis. So, once again, get everything ready. Alright, so this is the cap. I'm not going to take that off yet. Go ahead and pipette all that out. Into our alcohol. And we'll begin the process all over again. So I have my nutrient broth right here. TSA we don't need just yet. But nutrient broth for my Staphylococcus E. Then while I'm trying to get this to dry up for the sterilization process to take effect, we'll begin culturing the Staphylococcus E. Alright, so for our next one, I just washed my hands. We're going to start with the Staphylococcus epidermis, or epidermidis. So we're going to do this now. So start with the fresh pair of gloves. So a now sterile pipette. Start with our nutrient broth. So I've learned from my mistakes with the first one. So this time, few quick passes. Wasn't under the impression that these were plastic. So, careful not to touch the tip. We'll draw up 0.25. nutrient broth right there. This one is plastic, so I guess we're gonna have to be really careful. Definitely plastic. That's it. Still got a nice slight squeeze. Get all the nutrient broth in there. Once again, I shouldn't have done that. Should have just kept that. But sterilize it, dry it all over again. Recap this. Give it a good shaking and let it fully dissolve. So now we have our Staphylococcus E fully dissolved. We have our sterile pipette. We're now going to transfer it into the nutrient broth. So we'll just redo this one real quick. As well as this one. Should be enough. Squeezing. Now I believe these are going to be disposed of at the end of the exercise, but sterilize it just in case. Another cap. Re-sterilize before recapping. One came out far better. Now for our next part, we have our yeast, I believe that's Staphylococcus cerevisiae, S. cerevisiae. So that's the nutrient broth labeled S. cerevisiae. We're going to repeat the same steps. I have my half a teaspoon of my my yeast that I'll be adding to my warm tap water. Quarter cup, half teaspoon. So we'll go ahead and add that now. I'm gonna wait for that to froth up. And we're gonna repeat the same steps. Once that's all frothed up, after about 10 minutes, we'll draw up 0.25 from the yeast, and we'll add it to the S. cerevisiae nutrient broth. And we'll be done with this portion of the exercise. All right, now we're on to our last culture, which is the S. cerevisiae. So the yeast. The yeast is now frothy, a little foamy. So I'm gonna go ahead and grab our nutrient broth labeled S. cerevisiae. Well, that one's on there tight. Just flame it real quick. Sterilize the tip. Just a little bit. Now we're going to draw 0.25 of the yeast. Should be it right about there. Go ahead and put that in. By the way, the pipette was sterilized prior to drawing it. So there we go. It's in. We sterilize the pipette. Re-sterilize the tip. cap it. And there are our samples. And I believe these TSA deep, I believe this is the agar, the media that we're going to be using to do our plate pours. So I'm going to go ahead and begin those. These are the petri dishes ready for the plate pours. So that'll be next.